Hello everyone, and welcome. I'm excited to start today's presentation on a fascinating topic, body fluids and circulation. Today, we'll explore how our body fluids work and how circulation keeps everything running smoothly. We'll cover the key components of body fluids, the circulatory system, and their importance for our health. Let's dive in and learn together. Let's take a closer look at the functions of the circulatory system, which is the first topic on this slide. The circulatory system plays a vital role in maintaining the overall health of our body by transporting essential substances. It carries oxygen from the lungs to various tissues and organs, and it also transports nutrients absorbed from our digestive system to cells throughout the body. Additionally, it removes carbon dioxide and other waste products from tissues, directing them to the lungs, kidneys, or liver for elimination. The system helps distribute hormones that regulate various physiological processes and plays a key role in immune defense by circulating white blood cells. Understanding these functions highlights how integral the circulatory system is to our survival and well-being. As we progress, we'll explore specific components like blood, the heart, and blood vessels, each contributing to this complex but fascinating transport network. The circulatory system, also known as the cardiovascular system, is fundamentally about the movement of body fluids. Its main role is to distribute various substances to different parts of the body. Think of it as a transportation network that keeps everything running smoothly. It allows blood to circulate, carrying essential nutrients like electrolytes and amino acids, as well as oxygen and carbon dioxide. It also transports hormones and blood cells, which are all crucial for maintaining health. These substances are circulated to and from cells, ensuring the body gets nourished and functions properly. The key components of this system include the heart, which acts as the pump, and the blood itself, which carries all these substances. Additionally, we have the red and white blood cells, platelets, and the lymphatic system, all working together to keep our body in balance. Understanding this system helps us see how interconnected and vital our body's internal processes truly are. The circulatory system plays a crucial role in distributing water, H+, and various chemical substances throughout the entire body. This ensures that every cell and tissue receives what it needs to function properly. It also transports informational molecules, like hormones, from their sites of production to their target tissues, helping coordinate various physiological processes. Additionally, the blood carries metabolic intermediates, such as lactic acid, from muscles to the liver, where they can be further processed or oxidized. This highlights the system's role in managing byproducts of metabolism. It also facilitates the exchange of respiratory gases, oxygen from the lungs to tissues, and carbon dioxide from tissues back to the lungs, keeping our cells supplied with oxygen and removing waste gases. Beyond that, the circulatory system is responsible for transporting waste products from different tissues to organs designated for excretion, like the kidneys, ensuring toxins are removed efficiently. It also moves nutrients absorbed from the digestive system to various tissues and organs, where they can be stored, used for energy, or incorporated into new tissue components. Overall, these functions underscore how vital the circulatory system is for maintaining internal balance, supporting metabolism, and enabling communication within the body, all of which are essential for overall health and proper functioning. In this slide, we're focusing on the circulation of blood between the heart and the body organs. The key point here is that the left ventricle pumps oxygenated blood into the systemic arch, which then supplies it to the rest of the body through a network of arteries. This process is essential for delivering oxygen and nutrients to all tissues. Moving on, let's clarify the concept of systemic circulation. It involves blood traveling from the heart to the body and then back to the heart. The oxygen-rich blood is pushed out by the left ventricle into the systemic arch, and from there, it flows through various arteries to reach different organs. Now, let's distinguish this from pulmonary circulation, which is a bit different. It involves the movement of blood between the heart and the lungs. Here, the right ventricle pumps deoxygenated blood into the pulmonary aorta, which then ascends and divides into the right and left pulmonary arteries, leading to each lung. This is where blood becomes oxygenated again. Another important aspect is coronary circulation, which supplies blood to the heart muscle itself. The coronary arteries arise from the aortic arch just above the semilunar valves and branch into capillaries to deliver oxygen-rich blood to the heart wall. Meanwhile, the coronary veins collect deoxygenated blood from the heart wall and drain into the right atrium through the coronary sinus. Finally, it's worth noting that the physiology of blood circulation was first described by Sir William Harvey back in 1628. Understanding these three circuits, systemic, pulmonary, and coronary, is fundamental for appreciating how our circulatory system keeps every part of our body functioning properly. Blood is a type of connective tissue, which might not be immediately obvious, but it's actually one of the most vital ones in our body. It's composed of two main components, a fluid matrix called plasma and the formed elements. The plasma acts like a transportation highway, carrying nutrients, hormones, and waste products throughout the body. The formed elements include cells like red blood cells, white blood cells, and platelets, each playing a crucial role in functions like oxygen transport, immune response, and clotting. Understanding this structure helps us see how blood supports overall health and maintains our body's stability. It's an excellent example of how connective tissues are not just structural, but also functional in complex ways. Now let's talk about plasma, which is a crucial component of blood. The factors responsible for blood clotting are actually present in the plasma, but they exist in an inactive form. When these factors are activated during clotting, they help stop bleeding and form the blood clot. Interestingly, plasma without these clotting factors is called serum. Plasma isn't just about clotting factors. It also contains a variety of other important substances like glucose, amino acids, lipids, and more. These nutrients are essential for the body's metabolism and energy needs. In addition, plasma has small amounts of minerals such as sodium ions, calcium, magnesium, bicarbonate, and chloride ions. These minerals play vital roles in maintaining various physiological functions, including nerve transmission and muscle contraction. Now, focusing on the proteins in plasma, we find globulins, which are mainly involved in the body's defense mechanisms, helping fight infections. Albumins, on the other hand, are crucial for maintaining osmotic balance, ensuring that fluids stay properly distributed between blood vessels and tissues. Fibrinogen is another key plasma protein, and it plays an important role in blood coagulation, working alongside other clotting factors to form stable blood clots when needed. The major plasma proteins we should remember are fibrinogen, globulins, and albumins. Together, they make up a significant part of plasma's composition. Speaking of composition, plasma constitutes about 55% of the total blood volume. Around 90% of plasma is water, which acts as a solvent and medium for transporting substances. About 6 to 8% of plasma is made up of proteins, underscoring their importance in various physiological processes. Visually, plasma appears as a straw-colored, viscous fluid. Its color and consistency are typical features that help distinguish it from other blood components. In summary, plasma is a vital component of blood, carrying nutrients, minerals, and proteins essential for maintaining overall health and facilitating various bodily functions. The formed elements make up about 45% of our blood volume. These are the cellular components, including erythrocytes, leukocytes, and platelets. Collectively, we refer to these as the formed elements. They play crucial roles in transporting oxygen, fighting infections, and clotting blood. Understanding these components helps us see how blood functions beyond just a fluid, highlighting its complex and vital nature. RBCs, or red blood cells, play a crucial role in transporting respiratory gases throughout our body. They are specially designed for this task, which makes them vital for our overall health and function. The average lifespan of an RBC is about 120 days, after which they are broken down and destroyed mainly in the spleen. That's why we often call the spleen the graveyard of RBCs. The red color of blood comes from hemoglobin, an iron-containing protein complex that binds oxygen. In a healthy adult, each 100 milliliters of blood contains between 12 to 16 grams of hemoglobin, highlighting how abundant and important this protein is. Most mammals, including us, have RBCs that lack a nucleus, which allows more space for hemoglobin and enhances their oxygen-carrying capacity. These cells are uniquely biconcave in shape, which increases their surface area and flexibility, helping them navigate through tiny blood vessels smoothly. RBCs are produced in the red bone marrow, the primary site of hematopoiesis, or blood cell formation. Because they are so numerous, they are actually the most abundant cells in our blood. In a healthy adult, there are typically about 5 to 5.5 million RBCs per cubic millimeter of blood. Understanding the structure, function, and life cycle of RBCs helps us appreciate how essential they are in maintaining our body's oxygen supply and overall homeostasis. Let's take a closer look at the different types of white blood cells and their roles. Starting with basophils, these cells are known for secreting substances like histamine, serotonin, and heparin. These chemicals are crucial because they play a significant role in inflammatory reactions, helping the body respond to injury or infection. Moving on to lymphocytes, they make up about 20 to 25% of all white blood cells. There are two main types of lymphocytes, B cells and T cells. Both are essential for the immune response, working together to identify and attack pathogens, and they are central to our body's defense mechanism. Next, monocytes account for roughly 6 to 8% of WBCs. These cells, along with neutrophils, are phagocytic. They can engulf and digest microbes and debris, which is vital for clearing infections and maintaining tissue health. Eosinophils, comprising about 2 to 3% of white blood cells, are particularly interesting because they resist certain infections. They are also involved in allergic reactions, helping to manage the body's response to allergens. Neutrophils are the most abundant WBCs, making up about 60 to 65%. Their high numbers reflect their key role in the initial immune response, quickly attacking bacteria and fungi. Now, it's important to understand that white blood cells are categorized into two main groups: granulocytes, which include neutrophils, eosinophils, and basophils, and agranulocytes, which include lymphocytes and monocytes. This classification is based on the presence or absence of granules in their cytoplasm. Leukocytes, or white blood cells, are generally short-lived. In a healthy adult, we typically find about 6,000 to 8,000 WBCs per cubic millimeter of blood. Although they are fewer in number compared to red blood cells, they are nucleated and play a critical role in immune defense. Platelets are fascinating because they can release a variety of substances that play crucial roles in our blood. Most of these substances are directly involved in blood coagulation, which is essential for stopping bleeding and healing wounds. Typically, in just 1 cubic millimeter of blood, there are between 150,000 and 350,000 platelets, highlighting how abundant and vital they are. It's interesting to note that platelets are actually cell fragments, not whole cells, and they are produced from large cells in the bone marrow called megakaryocytes. These megakaryocytes are specialized cells that generate platelets through a fragmentation process. Sometimes you'll hear them called thrombocytes, which is just another name for the same thing. Overall, understanding platelets helps us appreciate their central role in maintaining our circulatory health and ensuring proper blood clotting when needed. Let's talk about the importance of blood groups, especially during blood transfusions. When someone needs a blood transfusion, it's crucial that the donor blood matches the recipient's blood type. If the blood types don't match properly, it can cause serious problems like clumping, which is the destruction of red blood cells. This can be dangerous, so compatibility is key. Now, there are some special blood groups worth noting. For example, blood group O is known as the universal donor because it can be given to anyone, regardless of their blood type. That's because it lacks the antigens that would cause a reaction in the recipient. Conversely, Someone with AB blood group is called the universal recipient because they can accept blood from all other groups without having a reaction. These concepts of blood compatibility are based on two major systems, the ABO system and the RH factor. These two groupings are used worldwide and are fundamental to safe blood transfusions. Understanding these groups helps us ensure that blood transfusions are safe and effective, which is a vital part of healthcare. Antibodies are actually proteins that our bodies produce in response to antigens. Now, antigens are chemicals that can trigger an immune response, meaning they alert our immune system to fight off what it perceives as foreign invaders. Interestingly, our plasma, which is the liquid part of blood, also contains two natural antibodies that are always present, even without prior exposure to specific antigens. Moving on to ABO grouping, this is a classification system based on the presence or absence of two specific surface antigens on red blood cells, which are called A and B. The way these antigens are expressed determines your blood type, whether you're a B, AB, or O. Understanding ABO grouping is crucial because it plays a key role in blood transfusions, ensuring compatibility and preventing adverse reactions. So, in essence, the presence or absence of these surface antigens on RBCs forms the basis of this important blood classification system. Let's talk about the significance of the RH group. If an Rh-negative person is exposed to Rh-positive blood, their immune system can recognize the Rh antigens as foreign and start producing specific antibodies against them. This is why it's crucial to match the Rh group before transfusions to prevent any adverse reactions. The Rh antigen itself is quite interesting because it's similar to one found in rhesus monkeys, which is how the name Rh came about. In humans, this antigen is present on the surface of red blood cells in about 80% of people. Those individuals are called Rh positive or Rh positive. Conversely, if a person doesn't have the Rh antigen on their red blood cells, they're referred to as Rh negative. Understanding this distinction is vital in transfusion medicine and pregnancy management, as it helps us prevent hemolytic reactions and complications. So, in summary, recognizing whether someone is Rh positive or negative is key to ensuring safe blood transfusions and overall health management. When a blood vessel is injured, the body quickly springs into action to prevent excessive blood loss. One of the key processes involved is blood coagulation. This process results in the formation of a clot, which is essentially a dark reddish-brown scum that forms over the injury site. The clot is made up of a network of threads called fibrins, which act like a mesh or a trap. These fibrins trap dead and damaged blood cells and other formed elements, helping to seal the wound. This entire process doesn't happen instantly. It takes a bit of time to develop fully. The formation of the clot is crucial because it acts as a temporary barrier, preventing blood loss and providing a foundation for healing. The mechanism behind this, known as coagulation, is an essential part of our body's defense system. It ensures that even in cases of trauma or injury, we minimize blood loss and set the stage for tissue repair. So, in summary, coagulation is our body's natural response to injury, forming a fibrin network that traps blood cells and creates a clot, effectively protecting us and promoting healing. When an injury occurs, it triggers a fascinating response in our blood. The platelets release specific factors that kickstart the coagulation process. This process is essentially a complex series of linked enzymatic reactions, meaning each step activates the next in a precise sequence. Most of these factors are present in our plasma in an inactive form, ready to be activated when needed. A key player here is an enzyme complex called thrombokinase. This enzyme is responsible for converting inactive prothrombin into thrombin, which is a crucial step in the process. Once thrombin is formed, it then acts on another plasma protein called fibrinogen, which is also inactive initially. Thrombin converts fibrinogen into active fibrin, which forms the mesh that stabilizes the blood clot. This entire process of blood clotting is essential for stopping bleeding and initiating healing. It's a beautifully coordinated system that ensures our blood clots only when necessary, preventing excessive bleeding while avoiding unwanted clot formation. Higher animals have a well-developed circulatory system that allows for very efficient transport of substances throughout the body. In these animals, the circulatory system is made up of a central pumping organ, which is the heart, along with various blood vessels such as arteries, veins, and capillaries. Arteries carry blood away from the heart to different tissues, while veins bring blood back from those tissues to the heart. This system is found in some invertebrates and all vertebrates, highlighting its importance across a wide range of animals. The discovery of the circulatory system is credited to William Harvey, who first demonstrated how blood circulates within the body. It's also interesting to note that there are two main types of blood vascular systems: the open circulatory system and the closed circulatory system. Understanding these differences helps us appreciate how various animals have adapted their circulatory processes to suit their needs. In a closed circulatory system, the blood flows through proper blood vessels, which are specifically named arteries, veins, and blood capillaries. Within the tissues, arteries divide into smaller branches called arterioles, and these arterioles further branch into tiny blood vessels known as capillaries. After the exchange of substances, capillaries unite to form venules, which then lead out of the tissues as veins. Arteries are characterized by their thick, elastic, and muscular walls, which are made up of three concentric layers, namely the tunica externa, tunica media, and tunica interna. All these layers contain smooth or involuntary muscles that help regulate blood flow and pressure. Capillaries, on the other hand, are extremely fine and thin blood vessels. Their walls are composed of a single layer of endothelial cells, which makes them highly permeable. They lack muscles and elastic fibers, allowing water, small molecules, and gases to pass easily between the blood and tissues. This exchange is essential for delivering nutrients, respiratory gases, and removing waste products. Now, in many advanced invertebrates such as prawns, insects, and mollusks, the blood doesn't stay confined within vessels. Instead, it flows freely through the body cavity and channels called lacunae and sinuses within the tissues. The body cavity in these organisms is called the hemicole, and the fluid circulating in it is known as hemolymph. In insects, for example, the tissues are directly in contact with the hemolymph, which circulates throughout the entire body thanks to the contractile activity of the heart. This type of system is known as an open circulatory system. So, to summarize, the key difference is that in a closed system, blood is confined within vessels, whereas in an open system, the blood or hemolymph bathes the tissues directly in the body cavity. Both systems have their own adaptations suited to the needs of different organisms. Let's take a closer look at the differences between open and closed circulatory systems. On the left, we see the open circulatory system. In this system, blood flows through large open spaces known as lacunae and sinuses, which are located among the tissues. This means that the tissues are in direct contact with the blood, allowing for a more relaxed and less efficient flow. Because of this setup, the blood flow tends to be quite slow, and the pressure within the blood is relatively low. This is sufficient for some organisms, but not as efficient for others. On the right, we have the closed circulatory system. Here, blood moves through a system of chambers, specifically the heart and blood vessels. This creates a sealed, more controlled environment for blood circulation. An important point to note is that in this system, the blood does not come into direct contact with the tissues. Instead, nutrients and gases pass through the thin walls of the capillaries into the tissue fluid, from which they reach the tissues. Because of this closed setup, blood flows more rapidly and with higher pressure, making it more efficient for transporting nutrients, gases, and waste products. Connecting this to our overall theme, understanding these differences helps us appreciate how various organisms have adapted their circulatory systems to meet their specific needs, whether it's for efficiency, energy conservation, or environmental adaptation. Let's take a closer look at the structure of veins. Structurally, veins resemble arteries quite a bit, but there are some important differences. The three layers that make up veins are very thin compared to arteries, and they are more elastic. In veins, the muscles and elastic connective tissues are poorly developed, which impacts their flexibility and strength. However, the collagen fibers in the outer layer are very well developed, providing some structural support. In most veins, the middle coat is extremely thin and contains practically no muscles at all. This is quite different from arteries, which have a thicker muscular layer to handle higher pressure. Another key feature of many veins is the presence of semilunar valves within their lumen. These valves are crucial because they ensure that blood flows only in one direction, towards the heart. They prevent backflow, especially in veins of the limbs where blood has to travel against gravity. Understanding these structural differences is important because they directly relate to how veins function within our circulatory system, complementing the overall theme of how blood vessels are specialized to perform their roles efficiently. The heart is the central pumping organ of the blood vascular system. It's a hollow, muscular structure made up of cardiac muscles that work tirelessly throughout our lives. This rhythmic activity keeps blood moving continuously, supplying oxygen and nutrients to all parts of the body. The heart is enclosed in a double-layered membrane called the pericardium, which is filled with a lubricating fluid known as pericardial fluid. This fluid reduces friction as the heart beats, allowing smooth, uninterrupted motion. Inside the heart, there are mainly two chambers. The first is the auricle, or atrium, which receives deoxygenated blood from various parts of the body. The second is the ventricle, which pumps oxygenated blood out to the rest of the body. It's interesting to note that the number of these chambers varies among different animals, reflecting adaptations to their specific circulatory needs. So, understanding the structure and function of the heart helps us appreciate how vital this organ is for sustaining life and maintaining overall health. Let's take a closer look at some examples like labio, scoliodon, and neoceratotus to understand their heart structure. The branchial heart in these fishes is quite interesting. It's thick, muscular, and made of cardiac muscles. It consists of two chambers, the auricle and the ventricle. This heart is often called a venous heart because it pumps deoxygenated blood to the gills for oxygenation. Once the blood gets oxygenated in the gills, it moves directly to the visceral organs, which means these fishes have a single-circuit circulation. Additionally, there is a sinus venosus and a conus arteriosus present, which help regulate blood flow. Now, if we look at lungfishes specifically, they have a slightly different heart structure with two auricles and one ventricle, which reflects their adaptation to both aquatic and terrestrial environments. Understanding these variations helps us appreciate the diversity in fish cardiovascular systems and their evolutionary significance. Let's take a closer look at the heart structure in amphibians. Starting with the basic components, we see that they have two auricles, which are the upper chambers of the heart. These auricles receive blood from different sources and help in pumping it to the ventricles. Now, moving to the ventricles, amphibians have an undivided ventricle, meaning the chamber isn't separated into right and left parts. This is quite different from the more complex hearts of mammals and birds. Because of this, their circulation is somewhat mixed, but still efficient enough for their metabolic needs. Speaking of circulation, amphibians have what's called double-circuit circulation. The right auricle receives deoxygenated blood from all the visceral organs via the precaval and postcaval veins. This blood then goes to the ventricle and is pumped to the lungs through the pulmonary artery for oxygenation. Once the blood gets oxygenated in the lungs, it returns to the left auricle via the pulmonary vein. From there, it moves into the ventricle again, completing the cycle. This dual pathway, deoxygenated blood to the lungs and oxygenated blood to the body, is what we refer to as double-circuit circulation. Now, let's look at some of the specialized structures. The sinus venosus plays a crucial role here. It's a collecting chamber that gathers deoxygenated blood from the body before it reaches the atria. Additionally, in cases like frogs and toads, we see the truncus arteriosus, which is the conus and the proximal part of the aorta. This structure helps in channeling blood from the ventricle to the arteries that lead to the lungs and the body. So, in summary, amphibians have a relatively simple but effective heart structure with two auricles, a single undivided ventricle, and a sinus venosus that helps in managing blood flow. Their double-circuit circulation allows for the separation of oxygenated and deoxygenated blood, which is vital for their survival in a semi-aquatic environment. Now let's take a closer look at the structure of the reptile heart. The conus arteriosus is divided into three main parts: the right systemic arch, the left systemic arch, and the pulmonary arch. This division is crucial because it helps direct blood flow efficiently to different parts of the body and lungs. Next, we see that in reptiles like crocodiles, gavials, and alligators, the ventricle is completely divided. This is an important distinction because, unlike in some other reptiles such as lizards, snakes, and turtles, this complete division allows for better separation of oxygenated and deoxygenated blood, supporting their more active lifestyles. Moving on, we have the sinus venosus, which acts as a collecting chamber for blood returning to the heart. It plays a key role in directing blood into the atria, ensuring smooth circulation. Finally, the heart also includes the left and right auricles, which are essentially the atria. These chambers receive blood from the body and lungs and are vital for maintaining effective blood flow. Altogether, these features highlight how reptile hearts are adapted for a semi-aquatic or terrestrial life, balancing between simplicity and efficiency in their circulatory system. Let's take a closer look at some key features of avian hearts. On this slide, you'll notice that in some cases, only the right systemic arch is present, which is an important aspect of avian cardiovascular anatomy. We also see the presence of both the left and right ventricles, which is typical for a complete and efficient circulatory system. For example, birds like pigeons have this setup, among other species. Another interesting point is that the sinus venosus and truncus arteriosus are absent in these hearts, indicating a more advanced stage of heart development compared to earlier vertebrates. And finally, there's a complete separation between the arterial and venous circulation, which ensures efficient oxygenation and nutrient delivery, crucial for the high metabolic demands of birds. All of these features highlight how avian hearts are specialized for their active lifestyles and high energy needs. Let's take a closer look at the key features of mammalian hearts. For example, in mammals like humans and rabbits, the heart has a specific structural organization. First, the heart has left and right auricles, which are the upper chambers. These auricles are responsible for receiving blood coming into the heart. Moving on, the heart also has left and right ventricles, which are the lower chambers. These ventricles pump blood out to the lungs and the rest of the body. One of the most important points is that there's complete separation of arterial and venous circulation. This means oxygen-rich blood and oxygen-poor blood are kept separate, allowing for more efficient oxygen delivery to tissues. Additionally, in these mammals, the sinus venosus and truncus arteriosus are absent. Instead, the heart structure is more advanced, with the systemic arch leaving the heart, which helps in direct distribution of blood. Overall, these features highlight how mammalian hearts are highly specialized for efficient circulation, supporting their high metabolic demands and active lifestyles. Let's take a closer look at the right auricle. It not only receives blood from the body, but also gets blood from the coronary veins, which drain blood from the heart muscles themselves. The right and left auricles are separated by a thin wall called the interauricular septum, ensuring that these chambers stay distinct. Similarly, the right and left ventricles are separated by the interventricular septum, which is a thick muscular wall. Now, deoxygenated blood from the body enters the right auricle through two main veins: the superior vena cava, bringing blood from the upper parts of the body, and the inferior vena cava, bringing blood from the lower parts. These venae cavae open directly into the right auricle because, unlike some other animals, the human heart doesn't have a sinus venosus. From the right auricle, the blood passes into the right ventricle through the atrioventricular aperture. This opening is guarded by the tricuspid valve, which has 3 flaps or cusps. This valve ensures that blood flows in one direction, from the right auricle to the right ventricle, and prevents backflow during contraction. Now, let's step back and look at the overall structure of the human heart. It's a four-chambered organ, consisting of two atria or auricles and two ventricles. This design allows for efficient separation of oxygenated and deoxygenated blood, which is essential for effective circulation. The heart is located in the thoracic cavity, nestled above the diaphragm, between the lungs. It's a muscular, hollow structure shaped roughly like a cone. In terms of size, it's about the size of a fist, around 12 centimeters long and 9 centimeters wide, and it weighs approximately 300 grams. Understanding these details helps us appreciate how the heart functions as a highly efficient pump, maintaining the flow of blood and ensuring that oxygen and nutrients reach every part of the body. Now, let's look at how oxygenated blood from the left ventricle is distributed throughout the body. It travels through the aorta, which is the main artery responsible for this distribution. The openings of the aorta and other major arteries are guarded by semilunar valves. These valves are crucial because they prevent the backflow of blood, ensuring that once the blood is pushed out, it doesn't flow back into the heart. Moving on, the left ventricle has some specialized structures called chordae tendineae and papillary muscles. These structures work together to keep the valves, both the bicuspid and tricuspid, closed during ventricular contraction. They prevent the valves from being pushed into the atria, which is essential for maintaining proper blood flow direction. Because the left ventricle does this important work of pumping blood to the entire body, its walls are thicker than those of the right ventricle. The increased thickness provides the strength needed to pump blood with enough force. Next, the oxygenated blood is pumped into the lungs via the pulmonary artery, where it gets oxygenated. After this process, the oxygen-rich blood returns to the heart through 4 pulmonary veins, bringing it back into the left atrium. From the left atrium, the oxygenated blood moves into the left ventricle through an opening called the atrioventricular aperture, which is regulated by the bicuspid, or mitral, valve. This valve has 2 flaps and ensures that the blood flows in the correct direction, preventing backflow into the atrium during ventricular contraction. So, in summary, this process highlights the heart's role in efficiently pumping oxygen-rich blood to the body and receiving deoxygenated blood back from the lungs, maintaining continuous circulation. Now, let's look at the key differences between the first and second heart sounds. First, the first heart sound, often called lub, is produced by the closure of the bicuspid and tricuspid valves at the very start of ventricular systole. This is when the ventricles contract to pump blood out. In contrast, the second heart sound, known as dup, happens when the semilunar valves close at the beginning of ventricular diastole, which is when the ventricles relax and fill with blood again. Another important point is the quality of these sounds. The first heart sound is low-pitched, less loud, and tends to last a bit longer, about 0.15 seconds. It's a softer, more prolonged sound. The second heart sound, on the other hand, is higher-pitched, louder, and sharper. It also has a shorter duration, lasting only about 0.1 seconds. When it comes to their frequencies, the first sound's principal frequencies are between 25 and 45 cycles per second, giving it that softer, deeper tone. The second sound's principal frequency is around 50 cycles per second, which makes it sound sharper and more distinct. Understanding these differences helps us better appreciate the mechanics of how the heart functions and how we can identify normal versus abnormal heart sounds during auscultation. Let's walk through the process of blood circulation through the heart. First, deoxygenated blood from the body drains into the right atrium via the superior and inferior vena cava, as well as the coronary sinus. Meanwhile, oxygenated blood from the lungs enters the left atrium through the pulmonary veins. This phase is part of the auricular circulation. Next, during atrial systole, about 30% of the blood passes into the ventricles, while the remaining 70% flows in during diastasis, which is a resting phase when the heart is relaxed. Then, as ventricular systole begins, the pressure in the ventricles rises. This increase in pressure forces oxygenated blood from the left ventricle into the systemic aorta, which supplies the entire body except the lungs. Simultaneously, deoxygenated blood from the right ventricle is pushed into the pulmonary aorta, which carries it to the lungs for oxygenation. Finally, the systemic arch distributes oxygenated blood to all body parts, while the pulmonary aorta directs deoxygenated blood to the lungs. This entire cycle ensures continuous and efficient circulation, vital for maintaining the body's function. The pulse rate is, therefore, the same as that of the heartbeat rate. This heartbeat rate, however, differs from species to species. In general, the smaller the animal, the faster its heartbeat. So, larger animals tend to have lower heart rates. For example, an elephant's normal heartbeat rate is about 25 times per minute, while a mouse's heartbeat can be several hundreds of times per minute. During each heartbeat, the blood is pumped from the ventricles of the heart into the aorta, which then distributes it to all parts of the body. This process occurs during ventricular systole and repeats approximately every 0.8 seconds. From the aorta, the blood flows into other arteries that supply different parts of the body. This movement causes a rhythmic contraction in the aorta and its main arteries, which we can feel as a pulse. This is what we call the arterial pulse. In a resting state, the average human heart beats about 70 times per minute. However, this rate can increase during physical activity, fever, or emotional responses like anger and fear. So, understanding the heartbeat and pulse not only helps us gauge health, but also reveals how the body responds to different conditions and activities. The heart receives nerve fibers from both the vagus nerve, which is part of the parasympathetic system, and the sympathetic nerves. The vagus nerve has an inhibitory effect, meaning that when it's stimulated, it slows down the heartbeat. On the other hand, the sympathetic nerves are acceleratory, so their stimulation speeds up the heartbeat. This regulation occurs because these nerves release specific chemicals, or hormones, when they are stimulated. Now, at the junction between the right atrium and the right ventricle, there is a specialized tissue called the auriculoventricular node, or AV node. This node is crucial because it picks up the wave of contraction that is initiated by the SA node. Sometimes, it's also referred to as the bundle of His. From this point, the electrical impulses spread through branches over the ventricles, forming what we call the Purkinje system. This network ensures that the wave of contraction spreads evenly across the ventricles, coordinating a strong, unified heartbeat. It's interesting to note that the mammalian heart is a myogenic heart, which means that the heartbeat originates from the muscle tissue itself. However, it's still regulated by nerves to adjust the rate as needed. Near the opening of the superior vena cava into the right atrium, there's a specialized muscle called the senoauricular node, or SA node. This is often called the pacemaker because it sets the rhythm of the heartbeat. The SA node is richly supplied with blood capillaries, highlighting its importance. The wave of contraction, or systole, begins from this node and then spreads throughout the entire heart, ensuring a coordinated and efficient pumping action. This automatic rhythmicity of the heart is essential because it allows the heart to beat regularly and adapt to the body's needs without conscious effort, maintaining the overall function of the circulatory system. Let's take a closer look at how blood flow is distributed to different organs, which we refer to as fractions of cardiac output. Starting with the cardiac fraction, it's about 200 milliliters per minute, which is the amount of blood that the heart itself receives. Next, we have the renal fraction, which is roughly 1,300 milliliters per minute, making up about 25% of the total blood flow. This highlights how vital the kidneys are in filtering blood and maintaining homeostasis. Moving on to the liver, the hepatic fraction receives around 1,500 milliliters per minute, accounting for about 28% of the blood flow. The liver is known as the busiest organ with a remarkable capacity for regeneration, which explains its high blood supply. Then we see the myofraction, which ranges between 600 and 900 milliliters per minute. This includes blood flow to muscles, and it can vary depending on activity levels. Finally, the cephalic organs, which cover the brain and the head, receive about 700 to 800 milliliters per minute. This ensures that the brain gets a consistent blood supply essential for its functions. Understanding these fractions helps us appreciate how the body prioritizes blood flow to different organs based on their needs and functions. The pumping action of the heart plays a crucial role in maintaining a pressure of blood in the arteries, which we call arterial blood pressure. This pressure is essential because it helps to pump blood at a high velocity along the arteries, ensuring that oxygen and nutrients reach all parts of the body efficiently. In a closed circulatory system, like ours, this pressure needs to be strong enough to keep the blood moving swiftly through the arteries. On the other hand, in an open circulatory system, found in some invertebrates, the blood pressure is much lower. This difference highlights how our body's design optimizes blood flow to support complex functions. So, understanding arterial blood pressure helps us appreciate how the heart works tirelessly to keep blood circulating effectively throughout our bodies. The blood pressure in veins is actually quite low, and that's mainly because blood has to pass through narrow arterioles and capillaries before it reaches the wider veins. This causes a drop in pressure, and at many points in the body, this pressure isn't enough to push the blood all the way back to the heart. Veins have thinner walls compared to arteries, which makes them more easily compressed. That's an important point because it influences how blood flows through them. To help ensure the blood moves in the right direction, veins are equipped with many valves. These valves are crucial because they allow blood to flow towards the heart and prevent it from flowing backward. Now, the movement of blood in the veins is also assisted by the contraction of muscles and changes in body posture. When muscles contract, they squeeze the veins, pushing the blood along. Similarly, when you change your posture, say, standing up or sitting down, it can help move the blood through the veins. And here's the key part. During these actions, the venous valves play a vital role because they only allow the blood to flow toward the heart. They prevent any backflow, ensuring that the blood keeps moving in the right direction. So, in essence, this process, muscle contractions, posture changes, and the function of valves, is a major way that blood is propelled back to the heart from the veins. It's a fascinating system that keeps blood circulating efficiently despite the low pressure in the veins. Let's take a closer look at the T wave. It indicates the repolarization that occurs during ventricular relaxation. This is when the ventricles are resetting their electrical state in preparation for the next heartbeat. Moving on to the QRS complex, this part of the ECG shows the spread of the impulse responsible for ventricular contraction. It starts from the atrioventricular node and travels through the bundle of His and Purkinje fibers, causing the ventricles to depolarize. Interestingly, this complex also reflects the repolarization of the sinoatrial node. The RS segment of the QRS wave and the SD interval illustrate the period of ventricular contraction, which lasts about 0.3 seconds. Essentially, QRS is directly related to ventricular systole, or the phase where the ventricles are actively contracting and pumping blood. Now, let's discuss the P wave. This wave indicates the impulse generated by the sinoatrial node and its spread across the atria, resulting in atrial depolarization. The interval between the P wave and the QRS complex, known as the PQ interval, represents atrial contraction and typically takes around 0.1 seconds. It's important to understand that these waves appear when depolarization has finished, and the muscle fibers are returning to their original polarity. That's repolarization. When both electrodes are in the same polarity, that is, both in a state of complete depolarization or repolarization, there's no deflection, and the wave drops down to the baseline. Repolarization waves, then, represent the generation of a potential difference. They only appear when the electrodes are in different fields, meaning one is depolarized and the other is repolarized. If both electrodes are in the same field, there's no deflection, and the wave remains at baseline. Finally, depolarization waves are associated with the active electrical changes in the heart muscle. Understanding these waves helps us interpret the overall electrical activity of the heart, which is essential for diagnosing various cardiac conditions. Some of the tissue fluid enters tiny channels called lymph vessels, and the fluid collected in them is called lymph. This system is known as the lymphatic system. These lymph vessels unite to form larger lymph vessels, which ultimately drain into two large lymph vessels called the thoracic duct and the right lymphatic duct. These open into veins, returning the lymph finally into venous blood, and thus it becomes part of the overall circulatory system. The movement of lymph is mainly driven by the squeezing action of the surrounding muscles. Because of this, the lymphatic system is relatively slow, and its flow can be uncertain. However, exercise can help increase the rate of lymph circulation. Generally, the rate at which lymph is formed matches the rate at which it returns to the bloodstream. But sometimes, the lymph formation exceeds its return, leading to an increased volume of fluid around the cells, which causes swelling known as dropsy or edema. Now, regarding the composition of this tissue fluid, it's very similar to plasma, but it contains much less protein. The reason is that some proteins are not filtered out through the capillary walls because these walls are impermeable to certain proteins. This tissue fluid plays a crucial role because it's the medium through which exchange occurs between the blood and the tissue cells. Through this fluid, materials like solids, liquids, and gases are transferred. Under the pressure of blood in the capillaries, some water and soluble substances are filtered out of the blood plasma into the tissue spaces, forming what we call tissue fluid. This fluid exists in the spaces between cells and a tissue and is known as interstitial fluid or tissue fluid. So, in summary, tissue fluid and lymph are key components of the body's transport system, facilitating nutrient delivery and waste removal, connecting the circulatory system to the tissue cells. Now let's look at how fats and lipids are transported in our body. When fats are absorbed in the small intestine, they don't just go directly into the bloodstream. Instead, they are packaged into a special droplet called a chylomicron. This chylomicron carries the absorbed fats and lipids from the small intestine to the blood, ensuring their safe and efficient transport. Moving on, I want to highlight the role of plasma proteins, which are large molecules synthesized by the liver. These proteins can't pass directly into the blood vessels because of their size, but they can diffuse into the lymph vessels through their walls. From there, they eventually make their way into the blood via the lymphatic system. And that brings us to an important function of the lymphatic system. It's responsible for returning interstitial fluid back into the bloodstream. This process helps maintain fluid balance in our tissues and prevents swelling or edema. So overall, the lymphatic system plays a crucial role not just in immunity, but also in fluid regulation and fat transport. Let's take a closer look at the lymphatic vessels. These vessels are quite numerous and are found throughout various parts of the body. They resemble veins in structure because they have all three layers: tunica externa, tunica media, and tunica interna. Additionally, like veins, they are equipped with valves, specifically semilunar or watch pocket valves, which help ensure the lymph flows in one direction. However, it's important to note that these valves are more numerous in lymphatic vessels than in veins, reflecting their role in maintaining unidirectional flow. Moving on to the lymphatic ducts or vessels themselves, these are small and thin-lined structures. They are lined with endothelial cells, resting on a basement membrane, and are quite delicate. Hence, they are described as fine. One end of these lymphatic capillaries is blind, meaning it ends blindly, while the other end unites to form larger lymphatic ducts that transport lymph throughout the body. Speaking of lymph, it can be best described as blood minus the red blood cells. It's a clear or slightly yellowish fluid that circulates within the lymphatic vessels. Think of it as a mobile connective tissue, similar to blood, formed by the filtration of blood plasma. This fluid plays a vital role in immune responses and in maintaining fluid balance within tissues. To tie everything together, the lymphatic system is essentially an extension of our circulatory system. It includes the lymph itself, as well as the lymph capillaries and ducts through which it travels. This system is crucial for immune defense, fluid regulation, and the removal of waste products from tissues, making it an integral part of overall health and homeostasis. let's take a closer look at the key differences between blood and lymph. Starting with the cellular components, red blood cells, or RBCs, are present in blood but absent in lymph. This is an important distinction because RBCs are responsible for carrying oxygen throughout the body. Now, when it comes to blood platelets again, they are present in blood but absent in lymph, which makes sense because platelets play a crucial role in blood clotting. Moving on to white blood cells, or WBCs, they are present in both fluids, but their numbers differ significantly. In blood, you'll typically see around 7,000 per cubic millimeter, whereas in lymph, the count can range from about 500 to 75,000 per cubic millimeter, reflecting their role in immune response. Both blood and lymph contain plasma, which is the fluid component, carrying nutrients, hormones, and waste products. The ratio of albumin to globulin proteins is also similar, greater albumin than globulin in both fluids, highlighting their roles in maintaining osmotic pressure and immune functions. However, fibrinogen, a key factor in blood clotting, is present in larger amounts in blood, but less so in lymph. This ties into the differences in coagulation properties. Blood has a stronger tendency to clot, while lymph's clotting ability is much less. Regarding the flow, blood moves in a two-way system, flowing from the heart to tissues and back, ensuring efficient circulation. In contrast, lymph flows in a one-way direction, from tissues back to the heart, helping in drainage and immune surveillance. The rate of flow is also faster in blood, facilitating quick transportation of gases, nutrients, and waste. Lymph moves more slowly, which is adequate for its role in immune response and waste removal. Finally, when considering the contents like glucose, urea, and carbon dioxide, blood carries less of these substances compared to lymph, where their concentrations are higher, especially urea and CO2, reflecting the metabolic waste that lymph transports from tissues to the bloodstream for excretion. Understanding these differences helps us appreciate how blood and lymph work together to maintain our body's homeostasis and immune defense. Thank you so much for your attention. Now, let's briefly review some of the key disorders of the circulatory system we've discussed. First, we have heart failure, which occurs when the heart isn't pumping blood effectively to other organs. This can cause a range of health issues because organs like the brain and kidneys rely on a steady blood supply. Next, we have angina, also known as angina pectoris. It's essentially an acute chest pain caused by a reduced oxygen supply to the heart muscle. Think of it as a warning sign that the heart isn't getting enough oxygen, often due to narrowed arteries. Then there's coronary artery disease, or CAD. This condition is caused by damage to the blood vessels of the heart tissues. It primarily results from the buildup of calcium, fat, cholesterol, and fibrous tissues inside the arteries, which narrows the lumen or the inside space of the arteries. This narrowing can restrict blood flow and lead to serious complications. Finally, we have high blood pressure, or hypertension. This is when blood pressure readings consistently exceed 120/80. Remember, 120 is the systolic or pumping pressure, and 80 is the diastolic or resting pressure. If readings reach 140/90 or higher, it indicates hypertension. Elevated blood pressure puts extra strain on the heart and blood vessels, increasing the risk of heart disease and damaging vital organs like the brain and kidneys. Understanding these conditions is crucial because they are interconnected and can significantly impact overall health. Managing these disorders effectively can help prevent more serious complications down the line.